Good evening, everybody. My name's David Hume. I'm director of the Roslin Institute. Thanks to all of you for coming out on this first wintry night of the year in Edinburgh. It's my pleasure tonight to introduce Professor Bruce Whitelaw, my colleague at the Institute. Bruce is a local lad, did his undergraduate degree in Edinburgh, and then went over to the dark side for a while, somewhere across the other side of the country, to work on transcriptional regulation as a postgraduate, and that started his interest in genetic control and genetic expression systems. He took a postdoc at what was then the AFRC's Animal Breeding Research Organisation at King's Buildings to work with the very new field of transgenic animals, and then joined the Roslin Institute, subsequently rose to become head of division, if that can be called rising. And he was involved in the very early animal bioreactor work in Edinburgh to make transgenic animals that produce therapeutic proteins, biomedical proteins in the milk of transgenic sheep. That work took a long time to come to fruition, but there are now products licensed from what one might call farming with a PH. He's worked ever since on large animals and been a pioneer in what we're going to hear about tonight. Bruce is a scientific advisor to a number of biotech companies, including Immunogenes and Recombinetics, and he works closely with the animal breeding industry, particularly with Genus, a very large pig and cattle breeding company, and with Hendrix Genetics, with whom the Institute just signed a collaborative agreement. Bruce has been actively involved in science communication for all of his time at the Roslin Institute. Is recently at the Cutting Edge Festival in Oslo and the Battle of Ideas in London, and he was also, actually not that recently, made a Fellow of the Royal Society of Biology. So it's a pleasure to introduce Bruce and ask him to tell us about genetically engineered livestock. Why?
Thank you, David, and obviously thank you to the organisers of this session of lectures, which I'm very pleased to be invited to give one to you tonight. But I just want to start off, I know it's dark, or it may not feel dark to you, but it certainly feels dark to me looking out to you. Could I just ask how many of you believe you know what a gene is? Could you just put your hand up if you believe you know what a gene is? Okay. If I said to those people that you had roughly 20,000 or so genes in your genetic makeup, would you put your hand up and say you agreed? A few, okay. So I now understand where I'm going to pitch this, because hopefully by the end of the night you'll understand a little bit more about some aspects of genes and also some aspects of the challenges and opportunities that we have with some of the wonderful technologies. I've put this word at the end here, partly to get you to come to hear me talk, but also to indicate that there are actually many ways we can use this technology, genetic engineering technology. And I'm going to labor tonight one of them, and that's within the agricultural context. So I am what's called a gene jockey or a genetic engineer. I'm enthralled by the ability to change pieces of DNA. Until recently, we were very crude at doing this. We put large chunks in, but now we can put in really quite small and even single bases, and I'll come on to that. So for those of you who've come to hear about the CRISPR revolution and the technologies and the opportunities that CRISPR technologies offer us, that will come. I will describe that, and I'll describe some of the work which we're doing at Roslin using these technologies. But first, I want to sort of set the scene, and this leads into why this CRISPR revolution is real. So the picture that's associated with this series of talks is this one here, and it touches on a number of aspects of our changing world. Pardon me. First of all, the fact that we are becoming much more urbanized. More and more of us are living in urban cities, and for those of us who have lived in Scotland and live in Scotland, this is difficult to know, but if you have the opportunity of travelling to some of the countries around the world where populations are much bigger, then you'll appreciate this. In my last trip to China, I visited 4 cities. The smallest of those cities had a bigger population than Scotland. We do not have a population issue here, but the world does. And that's shown in a graph like this. And many of you have seen this many times before. Many of you will be perfectly aware of the fact that we're roughly about here, and there's about 7 billion of us on the planet. And the estimate is that in the next 20, 30 years, that will rise to 9 or 10 billion. Now, most of you will have no idea what that size of number means. What does an increase in 2 billion, 3 billion people mean? Well, we live here. This is our city. And that 2 billion, 3 billion increase over the next 30 years, if it's translated to a daily basis, means that in the next 4 days, the equivalent of our city will appear on the planet. Within just over a month, the equivalent of our country will appear on the planet. And that's going to happen relentlessly for the next 20, 30 years. That's quite an incredible figure and offers many challenges. We have to house these people. We have to look after their health, both when they're young and when they're old. And our population age, the average age of our population around the world is increasing as we overcome many diseases, as we be able to treat many diseases, and as our ability to look after people gets greater. We need to educate these people. We need to educate them to a level where they can have employment and contribute to the cities and the worlds that they live in. A whole variety of employments, a whole variety of roles on the planet that we can offer. And we have to achieve all this. But we can't do any of that unless we feed them. Unless we provide the food, which allows them to be nourished, which allows them to grow, allows them to be healthy. With that comes a whole raft of aspects. And one aspect is, and it doesn't matter where you get your food, whether you're a subsistence farmer, whether you rely or involved in large-scale agriculture, whether this is your only animal or you own a whole herd of animals. It goes through some form of transport, some form of processing and transport, and we buy them. Some of us buy food out of supermarket wrapped up in cellophane, while others have the ability to buy food on the shelf, while others yet on our planet actually harvest the food themselves. It doesn't matter what part of that on our planet that your food comes from, you need this robust food supply chain. And the food supply chain is very complex, it's very diverse. There are whole aspects of it around the planet. This is obviously images which largely show a Western food supply chain, although these are animals in India. We have the animals themselves. We have what they eat, whether it be the grass or the forage for ruminants, or the grain or whatever other component we put into the food supply for them. We then have transport at some stage getting the product to some sort of processing, whether it be these nice jam tarts here or bottles of some component, whatever it might be. And again, there'll be transport to where you buy the food before you actually eat it. Luckily, at all these levels, we can intervene and improve our food production. And why is that important? Because we have to feed those 3 billion people, and we have to feed them from, how should I say, we cannot increase the amount of land particularly. We can do little tweaks around the edge of the amount of land that we can produce food on. But basically the amount of land that we can use to grow crops or to rear animals will decrease as we become more urbanized and as we use various plants for other uses such as biofuel. So we've got, in effect, a reducing amount of land to grow all this food, to go through this chain before it comes to our plate. The solution to this is multifold. And at all parts of this chain, we can intervene. We can improve the forage. We can improve the grain that goes into the foodstuffs. We can improve the animals themselves. We can look at how we transport food. Can we minimize that? Can we optimize that? And we can look at how we process food and finally get to us. So we do have a whole raft of opportunities to solve this challenge of gaining enough food for the planet. I'm going to focus on this, which is not surprising since I come from an animal genetics institute. I'm going to try and give you some description of how we can improve the genetics using some of the brand new technologies. It doesn't matter if it's a chicken, if it's a small ruminant, a large ruminant, a pig, a fish, or whatever other type of animal that is farmed. And we've been doing this improvement to animals for a long time. In fact, the whole process of domestication revolves around this. These are the cave paintings from France, and this is an artist's impression of what a village might look like 10,000 years ago. And we've got a few animals, some oxen here, some chickens, maybe some small animals and some pigs. And these were bred so that they could be co-housed with us. We looked for parts of their temperament which were beneficial for having within your close environment. We looked for animals that would live with each other. We didn't want them roaming around the prairie or the grassland that we lived in. And that worked really well for about 9,500 years. Then this chap came along. His name is Blakewell. He's from England, and he developed this animal here, which is one of the Lincoln sheep, traditionally Lincoln sheep. And what he did was quite a revolution. He started what we classify as proper breeding. Up until then, all this time and all the intervening time between that, animals were left to look after themselves. They were put in a pen, a cage, and a wood, or wherever it might be, and were allowed to randomly mate. And you would collect the animals that came out if you wanted to milk them, if you wanted to cull them to use their meat, and you took what you got. Now and again, there might be an infection or some other environmental stress put on the animals, which might select a few animals, which would affect the genetics in that population. But beyond that, they were left to look after themselves. This chap here had the brainwave of actually selecting which animals would mate. So he kept the boys and girls apart. And he would select specific male, specific female animals, in this case from the Lincoln sheep, to breed. And he selected on what came out of that. And that's a really huge step forward. It took 9,000 years to do that. Today's farming is a little bit more sophisticated than that. We understand genetics, we understand genes, we understand that there are differences between us. And I said at the beginning that we have roughly 20,000 genes. Well, we do. There's a school of thought that says we have about 50,000, and we could, aficionados amongst us in genetics might debate this at length. But we all have the same genes, or we have roughly the same genes. Certainly the vast majority of genes are the same between you and I. The difference is we have different alleles, we have different flavors of those genes, which means that some of us have got red hair, some of us have got not very much hair, some of us have got black hair. We may have different color of eyes, or some of us might be tall, some of us might be short. And that's because, not because we've got different genes, we haven't got the short gene or the tall gene. We have the same gene, but we have a different version of it, which we call an allele or a flavor. And we can identify these differences at the DNA level. And this most common, or a very common one, is called the single nucleotide polymorphism, where you have a single base change. And that forms a little flag, basically, on the genome, which we can identify using molecular tools. So we can use these to select specific animals. Those animals that were bigger might have this flag, while those animals which are smaller might have a different version or sequence at that point. And we can select that. And that was the basis of what was called marker-assisted selection. We now have gone to the stage where we can sequence the whole genome, so we don't have single tags. We have the full blueprint of the animal, which we can select on and breed on. And this has been really quite a revolution in the technology. What this allows us to do, either the marker-assisted or now the full genome selection, is pick animals which the top, this is a distribution curve of some trait within an animal, and we can pick animals which are the top end, and we can use those selectively using the same sort of idea that Blakewell introduced in the 18th century. Select from your best animals. He didn't know the best animals, he guessed the best animals, but we can now use genetics to tell us what we believe are the best animals. And with that, the next population will be better, however we define better. Geneticists call it genetic gain or genetic merit, economic merit if you're a little bit more financially orientated, but the animals are better. And in this way, over generations, the animal productivity is enhanced, and you get, rather than 1 bucket of milk, you get 2 buckets of milk. And we're really good at it. This graph here shows the increase in milk production in the dairy industry over this time period, over these 50 years. And you can see it slowly, this dips and troughs, but it slowly increases. We just go up and up and up. This one up here, the smaller one up here, is overall economic merit within the agricultural livestock system. And again, you can see there that the increase just gradually goes up and up and up. We're very good at it. So we've come quite a way from those ancient cavemen who are developed into small farming communities where it was just completely random, and you were looking to choose, and you looked, this animal's nicer for some reason. I perceive it's better, it's got the right color, it has the right plumage if it's a bird, and we chose that animal. And that, as I said, lasted on for nearly 9,000 years. We moved into the ability to identify specific markers, not very many, but specific markers, which we could then select on, and marker-assisted selection is that. And now we're at this stage where we're selecting on sequence. Now, there's subtleties to all this, which mean that this jump here is actually quite large and has been quite a revolution within agriculture. That's for a different talk. We are good at it. We can use this genetics, we can select. But we can only use genetics if the variation exists. So here's a ball pool. If you wanted a white ball, you are going to not find one. It's not there. You can only select, you can only choose animals with enhanced traits that you want if that genetic variation exists in the animals. So this is my very crude version of trying to teach genetics. The gene is for stripe. And Dad has got the horizontal stripe gene, and Mum has got the vertical stripe gene. And lo and behold, the young lad here has a bit from Mum, half comes from Mum, half comes from Dad. Now, I do apologize for any geneticists in the audience. You'll know that this is very, very crude. But the concept here is, here's Dad's got a black stripe gene, Mum's got a vertical stripe gene, and the boy's got the crisscross. If you wanted a red gene, or a blue gene stripe, or a yellow stripe, you can't get it. So as an analogy, here is a nice suburban street. And this family here have a Volkswagen Beetle, and it's orange. And in the morning, the chap can come out of this house, and he can choose, he can go in as orange Volkswagen, or his small hatch here to work. He can choose that. Imagine these are 2 genes. He can choose between those. His neighbor has got a blue Volkswagen Dormobile thing. This chap can't use that because it's not his car, and vice versa. This chap cannot use that. They've both got the Volkswagen Dormobile, but they can only use the one the color and the one that they own. So if we wanted this boy to have a yellow stripe, it just doesn't happen. Well, it does. It's called evolution, and that's why we're all different, and that's why we have this wonderful plethora of species on the planet. So we could wait. We could wait for evolution to give us the yellow stripe. And if we wait long enough, it will happen. We cannot predict when it would happen. It might happen tomorrow, or it might happen in 10,000 years. We can't predict it. A mutation in the gene that encodes black stripe would give us the yellow stripe. However, now we can do it in 1 generation, and we can do that using the tools called genome editors, which are these are hybrid molecular enzymes that will digest DNA at specific sites. And the second half of this presentation will go into this in some detail. So this is what I do, and this cartoon I stole off the internet shows you this chap here changing one of the bases. DNA is made up of 4 bases, A, C, T, and G. Whichever combination they're in encodes for the proteins that make up our cells. As I said, we've all got the same genes, we've all got the ACs and Ts and Gs, but in some parts of those genes you get subtle changes. One of us might have CCT, another person might have GCT or AAT. And those slight differences give us the different flavors, the alleles, which allow us to select. Well, these new genomic engineering tools, and I'll go into some of the terminology here just so that you're familiar with it, allow us to just do that, which is quite incredible. The first genome editing tool off the block was called the zinc finger nuclease, and it relies on an enzyme called an endonuclease, which will cut DNA. Now that enzyme is, in this case, it's called FOC1, and it has no specificity. It will randomly run up and down a piece of DNA and chop wherever it likes. So what the clever people in a company in California called Sangamo Biosciences did is they tied their nuclease, the DNA cutting enzyme, to a protein that recognized 3 bases. And that protein is called the zinc finger, hence the name zinc finger nuclease. They then put a few of these together, and this structure here allowing this series of protein domains to bind a very specific DNA sequence. As long as you have enough of these, you can bind a single sequence in everybody's genome, or any genome. You can have that level of specificity to hit the genome. This one is the next one that came along, and it's called the transcription activator-like effector nuclease, and it's exactly the same in concept, except that the protein domains are now called tails, or transcription activator-like effectors, and they bind a single base. But again, you can string them together to bind a whole bunch. The clever thing about both of these is you can tell the system in a test tube, basically, to deliver the combination of tails or the combination of zinc fingers to bind whatever sequence you want. There is probably not any single run of bases which cannot be targeted by one of these tools. The efficiency will differ between them, but every sequence can be targeted. This is the one that you've probably heard about, and it's called CRISPR-Cas9, and it's an even longer acronym, which is Clustered Regulatory Interspaced Short Palindromic Repeats. Cas9 is the nuclease. It's the equivalent of the FOC1 over here. The big difference, there's 2 big differences. The big difference is that this protein activity, the enzyme, is not tethered to the recognition component. The recognition component's an RNA, which goes in and recognizes a part of the target sequence, and then the Cas9 comes in and digests it. So what's special about these tools, apart from the ability to recognize any piece of DNA? Why are they so special, and how can we use them? Well, DNA damage, so they all do the same thing. They all cut DNA, double-strand break in DNA at a very specific site, and you can determine what that site is. DNA damage in the cell is bad news for the cell. It will trigger, unless it's repaired, it will trigger a cascade of events, molecular events in the cell which lead to destruction of the cell. When you develop disease like a cancer, which is an uncontrolled growth, the cancer cell has overcome the ability to be destroyed by recognizing DNA damage, because cancer cells are obviously often associated with DNA damage. So we have in our cells a whole raft of proteins that recognize DNA damage and fix it. And they're very good at it, otherwise we would not be sitting all here tonight together. They're very good at repairing that DNA damage, but they're not foolproof. They will make mistakes. So these enzymes come in, these genome editors, whether it's a zinc finger nuclease or a TALEN or a CRISPR-Cas9, will come in and will recognize the DNA and will cut. And if it cuts at that point, you get the DNA damage. These proteins come running in to rescue it and mends it. As soon as it's fixed, the editor will cut again. They come in, they fix it. And this goes on repeatedly, very quickly, until a mistake happens. Until there's a small deletion or a small insertion. And as soon as that happens, the editor will no longer cut, and the sequence will be changed. Now, this process is called non-homologous end joining, and is a way of creating a mutation at a very precise site The limitation there is you cannot control what the mutation is. It might be a small insertion, or a large insertion, or a small deletion, or a large deletion. If you add a DNA template into this mix, then these proteins, or part of the family of proteins, uses this DNA template to fix the repair. In doing so, you can change what was the original sequence to the sequence you put in. Now, if you remember, I described an allele, or these Volkswagens, the different colors. An allele is just having these single base changes or a small number of changes. You can change the allele of the gene. You don't change the gene, you can change the allele of the gene that you have. And you can do it in a cell, you can do it in a virus, you can do it in a bacterium, you can do it in a fungi, you can do it in a plant, you can do it in an animal, you could do it in us. So what are the types of mutations that happen in the evolution? Well, as I said, we have small disruptions, small insertions and deletions, which are associated with non-homologous end joining. You can have a large deletion, and you can have a modification, or, as I described, a change of the sequence of an allele. And these tools, these genome editors, allow us to do all of these, and to do them efficiently and easily. I should have said, for those of you who have ambitions or indeed are working with genome editors, the zinc fingers are a very difficult product to make. They have destroyed many postdocs' career. Don't do it. Get your £5,000 out, give it to Sigma or Sangamo, and they'll make it for you. The talons will cost you £300 to buy the reagents from a company called Adgene, which, if you're working in a molecular lab, you'll understand what that is. And they distribute reagents across the scientific community. And within a couple of weeks, you can be making your talons. When it comes to a CRISPR-Cas9, everyone in this audience could come into my lab tomorrow, design one, order it, down the wire. Next day, it will be delivered, and you can be putting it into cells. At Roslin, we've a number of projects, and I'll describe one in a minute to give an example of both the specificity and the power of these tools. But just to say, we've used it in an NHEJ configuration to create a premature stop codon, leading to a smaller protein, as shown on this protein gel here. We've used it to delete an exon or a protein domain from a gene, and we've used it to change the sequence to create a different allele of this thing. And we have pigs in our farm at Roslin which carry these genome-edited events, and sheep. I want to give you one example to try and illustrate the power of these things, because from a genetic perspective, those of you who have got an appreciation of genetics will have already realized all the breeding I told you about was within a population. If you want a Holstein Friesian, you breed with a Holstein Friesian. The genetic variation you have is in that population of animals. With genome editing tools, you can look for variation from any species and incorporate it into the animal of choice that you want. And this is what we've done in this project. We've taken what we believe is a genetic variation that exists within an African pig and introduce it into a Eurasian pig. And the reason for that is of this disease here, African swine fever. Now, it's a very severe disease within the pig species. This is the usual outcome of this. Either the animals die, and they die from basically bleeding to death within 7 to 10 days. Massive expansion of destruction of the inner organs. If the disease is identified, and it's, as I said, notifiable to the International Agency of Animal Health, the OIE, then there's a cull that goes on. It's quite a terrifying disease. It has the name Africa, and as you would expect, that is where it was originated from, in fact, Kenya. And it has a substantial limitation in sub-Saharan Africa, in that it prevents pigs from, or Eurasian pigs from being bred. However, it's no longer limited to Africa. And although we don't know exactly how or why, we believe a boat landed somewhere on the Black Sea coast here and some meat was either taken off the boat or disposed from the boat in some way. This virus can lie dormant in processed meat for up to a year. And something ate that meat, and it's now widespread in Western Russia, the old Soviet bloc countries, and Eastern Europe. It has a major economic impact on those countries. In fact, Russia predicted 3 years ago that its loss would be in nearly 300 million. But from an animal's perspective, or from a pig's perspective, it's devastating. If you get it, you're either going to die or your owner's going to kill you. There is no vaccine. There is no drug. That's not to say what the scientific communities are not trying to generate those. We are. But to date, there's no drug. And no, the only thing we do is cull them. Now, and I do apologize for those of you in the audience who are pig aficionados, but I'm going to briefly say there are 3 types of pigs on the planet. There's the very ancient Barbaroussa, which has lovely multiple tusks and is very hairy. Probably quite a scary thing to meet in a dark night, but apart from that, quite interesting. And they live in Indonesia, and we don't know much about them genetically. You have pigs that live in Africa, the bush pig, the warthog. And you have everything else, whether it's a large white, as shown here, landrace large white type animal, whether it's a pot-belly pig from Asia, whether it's a large wild boar or a mini pig or a micro pig. Now you can get pigs the size of cats. It doesn't matter. It falls into this category. If this virus infects one of these, this is the outcome. If this virus infects this animal, there's no clinical disease. So the virus is the same, the genetics must be different. Now, clever people down at the Institute of Animal Health, now called the Purby Institute, Linda Dixon in particular, was looking at the virus, and viruses are very clever things. They encode proteins which suppress or evade our immune system. And one of the proteins goes by the really exciting name A238L. And it looks like a transcription factor found in the nucleus of animal cells. That transcription factor, or it looks like the inhibitor of that transcription factor. It looks like a protein called I-kappa B, which regulates a transcription factor called NF-kappa B. NF-kappa B is one of the transcription factors that controls the expression of cytokines, and it's a cytokine storm, an uncontrolled cytokine burst that kills the pigs. So here we have a viral protein which looks like a suppressor of the pathway which actually leads to the death of this type of pig. So I set a student the task of sequencing the genes in this pathway here. And he sequenced a couple and came to me and said, Bruce, they're exactly the same. This is a rubbish project. He then came back with another two and he said, these are also exactly the same. This is a really rubbish project. Right? And this went on for about 6-9 months until he hit what we think might be the jackpot. It's a gene called REL-A, encodes P65. It is one of the major components of NF-kappa B, shown here. And what we found was that there were 3 amino acid differences in the warthog REL-A versus the domestic pig. And this one in particular is a phosphorylation site that we know regulates the activity of this protein. Now, you may be saying, well, why don't you just take this animal and mate it to this animal? Well, they may look like pigs to you, but these are different species, and species barrier means you can't cross them. This animal is genetically as close to this animal as we are to marmosets. So the simple solution of mating them is not possible. We looked hard within populations of this to find this sequence and failed to find it. Doesn't mean it's not there, but we've just not found it yet. So here's a really good possibility that we could, or project where we could use the genome editors to take this sequence, incorporate it into this animal, this gene here, and ask whether this animal was still susceptible to the virus. I don't have the punchline, but I can tell you that we've done the genetic bit. We have used the option of a template to change these 3 amino acids in the protein by changing the underlying DNA sequence. And these animals are being bred at Roslin and will be challenged with the virus to see if they are actually less susceptible to African swine fever. There is no alternative way of changing the genetics of this type of animal to this sequence beyond waiting for evolution to do it. A lot on this slide, don't pay attention to that, just to say that this approach to try and alter the genetics of the animal using these tools can prove very productive. There are 4 different graphs here. This is the control animal, this is the edited animal. There's no symptoms whatsoever. And the same here, that's the control animal, that's the control animal, and along the bottom are the symptoms. This is a disease called PRRS, or porcine reproductive and respiratory syndrome virus, caused by the PRRS virus. And again, it leads to, in many cases, death of the pig. It is the single biggest viral burden within the pig industry. And by editing a specific gene, it's called CD163, colleagues in the States have managed to generate animals which are resistant to the virus. So we're not quite yet there with our ASF project, but this shows you the power of how these tools can be. Again, the change that they've engineered into these pigs is not one that is normally found within the pig population. Okay, just to finish up with a few more slides. And our changing world, the title of this series. Well, I want to get across to you tonight that there are 2 things which are changing in our world. Food, the need for food, the need to improve our productivity on the farm because we have a lot of people to feed, and every day we have a lot more. But also the fact that we have what is sometimes called the CRISPR Revolution, the genome editing revolution. We can literally go into a genome and change a base. We can take this G and make it an A. We could make it a C. We could make it a T. We could do exactly the same to the base beside it if we wanted. There isn't any piece of genome, any animal, any virus, any bacteria, any plant that we can't do that to. And what's really remarkable, and if any of you in the audience are working with these tools, is it's relatively easy to do. It's not simple to do. It's relatively easy to do. There will not be many molecular labs around the world and universities which are not using these tools. So we can change the genome. The question is, why do it? I've illustrated tonight the possibility of using it to enhance disease resistance in animals. And we at Roslin believe this is a very powerful and appropriate use of this technology. I would emphasize it's only one part of the solution. If we go back to this food chain, which we need to develop, we can improve all the way through here. Some of this will take high tech, some of it will take low tech. And it will depend on where in the world you are and the geography and the environment that you work in. But the genome editing tools offer us part, or can be part of the solution to that, addressing here. We could also use them to improve the forage and feed going into the animals, if that's what we wanted to do. So I'll just finish up by saying I get the opportunity of standing up here and talking to you, but I work with a team of people back at the Institute, some of which I'm pleased to say are actually in this audience, so no hard questions from them. Obviously, we work with animals, and our farm staff look after them extremely ably. And we work with a number of funders. Our primary funder is the BBSRC from the government, and also Innovate UK, which some of you may know as originally knew as the Technology Strategy Board. But we also work with industry to get access to these cutting-edge technologies and to use them in projects which we believe and industry believes are useful. And I'll finish with one last slide. And that's these 4 animals here. Now, they all have one thing in common. They all have a mutation in the myostatin gene. Now, the myostatin gene gives you extra muscle. As you can see in some of these animals, they are quite muscular. 2 of them are natural mutations, which we have in our fields today. And if you drive out into the country, you'll see at least some of these. And 2 of them have been created using the genome editing technology. I challenge you to work out which is which. Thank you for your attention.
It's been a minute since I've learned about genes. Are viruses Is it just DNA, just RNA, or neither? This is going to go into my question.
A virus.
Yeah.
The virus itself, we have two types of viruses that have DNA and viruses that have RNA.
Okay, so is it possible to basically use this technology to get into a virus and then alter their dormancy?
Potentially, yes.
Okay, so if that's possible, rather than trying to go after an immediate change in basically a single turn of changing their genetics, is it possible to just like make them dormant to life expectancy or something so that it'll always have it and then just never feel the effects of it?
So I think correct me if I'm wrong, I think the question is, should we engineer the virus to prevent it from being nasty? Yeah. Yes, we can. And I would then ask you how you distribute that out to the world. All right, so you're going to make a virus that doesn't replicate, let's say. What can you do with it? You can't actually get it out there. So, but yes, you can, and there are many projects looking at using these tools and other similar tools to mutate the virus and work out which bits are important. But distributing that virus out requires, by definition, the virus is replicative, and that's challenging. So, but yes, you could. You could try and do that and work out ways. And the equivalent is being trialed in insects. You may be aware of trials that are going on just now in Florida and Brazil, looking by a company called Oxitec, which are trying to distribute male sterile mosquitoes to try and impact on malaria. And although the ones that have been trialed have not been generated using these tools per se, the next generation ones have. And that's a similar type of idea, whether you could do that. And there are other more complex ways of trying to do it, including a technology which is highly controversial called gene drive.
Thank you.
So Bruce, what do you think we shouldn't do in editing? Where does the boundary?
Where's the boundary of editing? I think we have to be extremely careful when we start to edit viruses to make them change their tropism. I think we have to be very careful if we edit viruses to enhance their ability to overcome our immune defense. And to a certain extent, that applies to bacteria as well. The sort of elephant in the room is, do we use this on ourselves? And there are already preclinical trials of using genome editors to treat disease, which can either be called cell therapy or gene therapy, depending on how you look at it. And in that context, the ability of the genome editor itself doesn't affect our ethical or our risk management of that. Where the new technology kicks in is the fact that, so what I didn't explain is how we do this at Roslin is we take the genome editor, be it an RNA or a protein, and we inject it into the fertile egg, the fertilized egg. We then put that fertilized egg back into the animal, and in the majority of cases, that egg will develop into an animal, and we'll have, in some of those cases, something edited. Well, we could do exactly the same to the human fertilized egg, if that's what a society deemed was useful. There's a huge public debate about this, and a number of summits and organizations have come together. And if I paraphrase the outcome of it, it's not yet. That's not no, it's not yes, but it's not yet. I think we'd have to think very hard about how you could justify using this technology to alter the human germline.
One of the things when you think about sustainable food supply into the future is that it takes a lot more energy to produce livestock as opposed to grains, et cetera, just because of the inefficiency of transferring the energy up the food chain. But some animals are more efficient than others. Like, they tell you to eat less beef and eat more chicken or whatever. Is there a possibility for essentially making cattle that would have a better transfer efficiency so that you wouldn't need so much land to grow food to make cattle, and therefore people could continue consuming meat while also having more sustainable agriculture?
So, absolutely. If you think about it, if you have 10 animals and a disease comes through and kills 5 of them, that's an incredible waste. if you had those 10 animals and they were resistant to the disease, that would already be an efficiency. And that's certainly how we view one of the outputs of looking at disease resistance. There are also other aspects of our farming practices which have huge wastage attached to them, and we could look at how we do that to make it more efficient. And the one that jumps out is in the layer industry, the chicken layer industry, or the bovine dairy industry. The need for a male Holstein Friesian is limited because it doesn't produce milk, and exactly the same for the need for a male layer chicken, which doesn't lay eggs. Currently, in both situations, it's roughly 50-50 offspring, boys and girls. And for those of you who don't know, in the layer industry, one-day-old chicks are killed. The lucky ones get their heads, necks broken. The unlucky ones will be thrown into an incinerator or some other macerating machine. We could use this technology to what we call gender skew. So you could bias the production of one sex or another in the offspring, and that would have a major impact on the overall productivity within that scenario. We can also look at breeds where we feed animal feed to breeds, or fish in particular that eat meat to grow, and that's an incredible inefficiency in the system, and we can look to ways to improve the efficiency along those lines too. So yes, we can have an impact on that. Another aspect of your question is, should we keep eating meat? And so this population rise that's shown here, what's not often talked about is the expectation it'll plateau off about 9 or 10 billion. And the majority of predictions imagine that's going to be the case in about 30 years' time. Now, many of you in the audience, especially those of you who are at the university studying here, will have the pleasure of seeing that moment. And that environment, if I was able to live that long, that environment, I would like to see both meat and plant products on the table. So where genetics comes in and what gives us a real advantage is that we can stably change a population for the better, be it increased productivity or disease resistance. And really this technology, yes, it'll help us get to that point, but where it really comes in is after that, where I suspect society will still want a mixed diet of one sort or another.
There was an article in the New York Times last week, and there was this argument that since genetic modification was introduced about 20 years ago, there has been no appreciable difference in the amount of food yield in America, where it is practiced, and where GM crops are grown, and in Europe, where it is banned. And so there might be those that would argue against the introduction of this type of technology for animal breeding. I mean, how do you convince anyone that GE will actually improve the number of animals we produce in the future? And my second question is, how do you actually intend to introduce genetically gene-edited animals into the production system? Do you replace all the currently producing animals, or I don't know?
Okay, so two questions there. I think the first question is really, how do you convince, if that was what you used, I can't remember if you used that word or not. How do you portray arguments that make this, well, the two examples I just gave, disease resistance and reducing the number of needless deaths within the industry, I think are strong arguments. And if we can generate edited animals that have that type of trait, then I think it's something society has to look at. The debate that we hear in the public about the pros and cons of this and the risks and so forth is highly colored by a variety of things, and it's very difficult to actually get down to the truth of it. And from one perspective, people say that the amount of GM crops that have been grown in the US haven't changed things, but on the other hand, 70%, and sometimes even more than that, of most crops in the US are now GM. So it is an ongoing debate. How do you do it? Well, you stand up in front of an audience you don't know, and well, I happen to know you, but apart from that, you stand up to an audience you don't know, and you respond to questions. I think these technologies need to have really good examples, and the one I briefly touched on there about the PRRS-resistant pigs, I think is a fantastic example. There are 100 million pigs alone in the US susceptible to this virus. Costs the country $600 million a year. That's an economic value. All those 100 million pigs theoretically could die of this virus. That's a welfare issue. The second question is, I forgot it. I'll go briefly. How do you introduce it? All right, again, this depends. In the Western agricultural system, where we have elite animals and we have what we call multiplier facilities underneath, where you increase the number of those animals before they're sent out to the farm itself, it'd be very easy to integrate into either just below the elite or into the multiplier. All right. Now, I know you're from Nigeria, and I know the situation there is somewhat different. It will be challenging to get it out into the small farming communities that you're more familiar with in Africa. And the challenge can be overcome. In the Western world, it's going to be industry that promotes that for a beneficial trait like PRRS. In a country like yours, we need government input and government ability to spread out. But in effect, the same way can happen. We could have government-based multipliers that distribute out selectively to the farmer. It won't be as quick, but it can be done.
I think as an interesting aside, the assertion that the productivity hasn't increased is just plain wrong. And if it hadn't, I don't think the farmers would be planting genetically modified crops on the scale that they do. There is, in fact, a massive saving. But the interesting thing, which I think was said by a previous speaker in this series, one forgets that the people who introduced genetically modified crops were, in fact, environmentalists and who were concerned about polluting the planet with herbicides and pesticides. And the use of pesticides and herbicides has been drastically reduced. So I think one can portray these stories in rather different ways.
Would it be possible to change, I don't know, an organism like an algae into producing energy? And then the second one was, would it be possible to maybe, like, in the near future, be able to inject, I don't know, mosquitoes or another organism to be able to produce or spread antibiotics or vaccines or something like that rather than infection? Would it be possible at all?
Inject an insect to be able to secrete a vaccine. Possibly, we could imagine ways where it could produce a protein that once in the
It's a good idea. We should write that one down.
Possibly, yes. David's right. It's triggering lots of thoughts in a minute. So yes, we could. There is that's the wrong way to put it. There is a study going on just now where mosquitoes have been infected with a specific bacteria which seems to affect the immune system or the ability of the virus to replicate. So there are attempts in that general area. But yes, you could get the bacteria or the virus to a certain extent to have some protein production which could affect the immune response of the animal in some way or other. Yes, you could. Obviously, you could do it the other way around. You could get the virus or the bug to secrete something which is very nasty. And again, that's where I hope regulations come in to help us. And sorry, your first question was?
Be able to produce organisms like algae.
Oh, yes, for energy. So, yes, if we know enough about the pathways involved in producing whatever energy source there is, and, don't get confused, it's not electricity, light, there's a whole variety of ways that energy can be produced, and we then have to have a way of capturing it, which could be challenging. The ability to change a series of genes or a pathway itself or introduce novel pathways has certainly been looked at in research environments. So again, yes, that is possible. Not to say it's simple, but it's possible.
I think I have a long question that I've written. Just wanted to ask, is it not possible to just manipulate the genes rather than inserting a gene in an animal? Because you did say that if you insert a gene, you would need to wait for the evolution process to take place so that you would see the effects.
Okay, so I may have not made it clear. You rely on the proteins within the body, which is evolutionary conserved proteins in the cell, but the effect is in a single generation, so your offspring carry the change. Certainly with the plethora of biobanks we have across a number of species around the planet, we can certainly use these tools on those cells. And in fact, if you were to genetically engineer a chicken or a bird, you would use a cell-based system, and it's just in the large animals, the ruminants and farm animals, and in experiment animals like mice and rats, that we can get easy access to the fertilized egg that we use the egg. So yes, you can engineer variation into biobanks. You've then got to do something with the biobank, but the actual effect of these editing tools is realized in the first generation. And it's then stuck. It's inherited in a Mendelian manner from then on.
Because you said it's very possible to change or alter any gene or any specific base on a gene, how can we as the audience or as the nation be assured that someone is not going to wake up and just manipulate genes and change the whole system altogether just because they can?
Well, to be absolutely honest, we can't, do we? But I was given this question on a different point when somebody described that he had heard about these tools a couple of years ago while I was working in a lab in Paris, and his first thought was that we're doomed. And my answer to him and my answer to you is the same, in that I have a much greater faith in society than he did. I'm sure you do it as well, and that we regulate and we police it. That's not to say we'll never ever have things which break the law. The challenge we have at the minute for most countries around the world is that there's not clear legislation for the use of this technology either in plants or in animals, although the plant situation is much more advanced. That's the same for the release of insects or bacteria or viruses. We have a random process going on just now where some countries are deregulating it and other countries are looking to capture the regulations under the GMO, and Europe's perhaps one of those. So my answer to you is there's a real risk that this technology could be used badly, but then there's a risk that any technology be used badly. We don't ban things because necessarily that they have no risk attached to them. We try and manage that risk. If we were to ban things that carried a risk, we'd have all had to walk here tonight, and we would not indulge in a pint of beer or something like that later. And indeed, we would have a substantially limited food intake than we currently do. So the solution is to manage it. The solution is to have regulatory systems in place and to have public debate about how we get to that position.
Hi. I was just wondering if is there theoretically a solution to any disease somewhere? You know, like pine trees don't suffer from ash dieback and sheep don't suffer from bovine TB.
Yeah, theoretically, yes. And that all these diseases, be it a virus or some sort of pathogen, has to gain access to the animal and then has to be able to find a niche and replicate. In the case of a virus, that's infecting a cell. In the case of a bacterium or some other protozoan or some other pathogen like that, they'll find a niche that they can hide and survive. So if you could block that ability, in theory, you could stop any disease, infectious disease. In the case of genetic disease, well, that then gets back to the question of whether how you do that within a human context. But theoretically, yes, you could prevent any disease. There's also been the long-term hope of many of the scientists that work within specifically viral diseases, the fact that we could find the gene that stops all viruses or something. I think that's perhaps a taller order, but there may be ways that we could enhance the immune response of an animal, or indeed suppress the immune response of an animal, as in our REL-A case, which is a suppression, which would then allow the animal to be able to resist a wider spectrum of diseases. So that's possible. We don't know enough about the biology to make that a reality today.
Yeah, I was just wondering about the variety of species on the planet, because the more humans we have and the species that we grow for food, both animals and plants, are dominating in the world. And I was just wondering, does genetic modification of farm animals have any effect at all on the speed that wild species are dying?
There will be circumstances where you have a farming environment which impacts on the neighboring and local population of animals. But genetic engineering is not necessarily going to make that any different from today. But there are theoretical questions there which need to be answered. If we take foot and mouth disease, for example, which is a virus which, when it has an outbreak in the United Kingdom, we get particularly hit up about, and has a major impact on both economics and society here. If you were able to generate livestock, so foot and mouth disease infects a whole range of animals. It unfortunately infects livestock and therefore affects the food we eat. But the bigger number of animals is probably in the wild. So you could imagine if you actually impacted on the prevalence of the virus in a farming environment, you could actually have a beneficial effect on the outside animals, the wild animals. So in theory, you could have indirect beneficial effects. Given the fact that these are tiny little molecules, there's nothing to stop the future, not today because we don't have this technology, but it's not far away. There's nothing to stop us adding those small genome editing molecules to some vector, be it a piece of plastic or some vector, and distributing it into wild populations. So you could actually see this genome editing technology have an impact, and direct impact in wild populations. Now, I'm not aware of anybody doing that at the minute, and I don't see where the financial driver for that will come at the minute. But so you could see it being used, or seeing an impact in both ways. The fact that we can genome edit livestock in the agricultural system just gives more variation for the farmer and the breeder to select on. It doesn't change any of the other parameters until we move to disease resistance, which then could have beneficial knock-on effects.
Given that the majority, if not all, of current domestic species or domestic breeds have been domesticated a long time ago, I'm not aware of there being another wild animal that's gone into domesticity and mass food production in recent times.
Ostriches.
Ostriches, to some degree. Are we therefore tinkering with the product of many centuries of deselect or artificial selection and minimization of domesticated animal gene pool? Is it time, in conjunction with what you're doing, to start again, using the warthog example or something of that like, where we go back to wild animal founders within domestic populations and start to construct domesticated animals for future needs rather than playing with breeds of sheep that have been around for perhaps too long?
So I think going back to the wild version is probably a bit challenging. Our ability to, so there was one slide there which I glossed over very quickly, describing how we can sequence genomes. Our ability to sequence DNA is incredible nowadays. It's cheap, it's quick, and based on that, we can gain an awful lot of genetic information. And much of the activity that here in Edinburgh we're involved in, and Rosalind in particular is involved in, is reaching out to other parts of the world which have a different genetic diversity to what we have here. And combine the sequencing power and the genome editing power, you can get to a situation where we can actually incorporate them in a controlled manner, like the warthog example. to go back to the original warthog and to make it that, and so the or that breed of warthog that you saw there is about half the size, physically half the size of one of our large white animals at an equivalent age. It would be a long route to go, I think, going back to the wild animal. It's possible. A colleague of mine, ours at Roslin, gave a talk once, and it was talking about extinction and how we can get involved with that. So that led me thinking, and I have another slide I didn't show today, which has the wolf and all the different breeds of dogs we have today. Well, you could take the wolf, today's wolf, and using Eddington, you could design your pet of choice, or conversely, you could take your Chihuahua and you could edit it back to being a wolf.
I guess that's been tested with the silver fox experiments, where they selected foxes for domestication. I guess might wind up there. I think this area has become incredibly topical over the past couple of years, so much so that the term Nature recently coined the term biohacking for backyard genome editing. It really can be done in a backyard. It could potentially be done on the farm with a 4-cell egg, which you'd cut into 4 and genome edit each one individually. So the potential is extraordinary. The opportunities are extraordinary, and I think just like to finish by thanking Bruce for giving us the insights into where we're going and the opportunities to solve some of the world's problems. Thanks, Bruce.